we have looked at various mechanisms of catalysis by enzymes. So, we first looked at approximation, in which the substrates, the two reacting molecules are brought close to one another and in a very preferential manner, so that the reaction can occur. And then we looked at covalent catalysis, where an intermediate, a highly reactive covalent intermediate is produced during the reaction, which results in acceleration. And subsequently, we looked at acid base catalysis, where there is a transfer of a proton or a hydroxide ion, and that results in generation of reactive intermediates or acceleration. There are broadly three other ways in which enzymes are able to catalyze reactions. Next mechanism by which it does it is electrostatic catalysis. Okay. So, in electrostatic catalysis, what happens is that the transition state that is produced during a reaction is actually stabilized by residues in the active site. Okay. So, here the transition state will most likely involve the generation of an ionic charge or a partial ionic charge. So, for example, when you have a negative charge that is being produced here, the incipient negative charge can be stabilized by a residue that is positively charged. Okay. So, fundamentally this form of catalysis involves electrostatics and stabilization of the transition state by an oppositely charged species. Okay. Next, we will look at a mechanism called as desolvation. So, desolvation, basically the concept of desolvation is that many reactions that are much faster in the gas phase when compared to the solution phase. So, as a concept, if you want to understand this, if there are two molecules that have to collide to react, when there is a solvent surrounding it, it has to displace the solvent and then collide. Whereas, in gas phase, it still has to displace some gas, but because of the easy fluid nature of the gas, you do not have that level of resistance. So, an enzyme active site is more or less devoid of water. Okay. So, in this it is usually a hydrophobic cavity, and therefore, there is very limited amount of water. So, in this situation, when the molecules are getting into the active site, it is almost like a gas phase situation. Okay. So, this concept is known as desolvation, wherein the solvent molecules, in this case water, they are removed from the active site, and that facilitates the reaction. Okay. But conceptually, if you want to look at it, what essentially happens is that once the molecules are getting in, and if the molecules are polar or charged, when water molecules are removed from these polar or charged groups that are getting into the active site, what happens is that there is a increase in the ground state energy. Okay. So, you may recall that rate acceleration occurs when the transition state goes down or the ground state goes up, and of course, when both of them happen, there is going to be a large increase in the reaction rate. So, here ground state destabilization largely contributes to an increased rate. Okay. Now, to understand this, we also need to understand dielectric constant, because dielectric constant is nothing but the ability of the medium to separate charges. So, water, which has a very high dielectric constant, if you remove it from the active site, then it is likely that the molecules that are getting in are going to experience a completely different dielectric environment. So, together these contribute to ground state destabilization. Now, another view of this is that instead of thinking about it like as being a gas phase, it is perhaps more appropriate to view this mechanism as solvent substitution. Okay. So, when you have an active site, and inside the active site there are groups that are around in the active site, which can sort of solvate the substrate. Okay. So, these provide specific polar environments in the area, and so instead of thinking about a gas phase, we can instead think about solvent substitution, in this case substitution by a polar environment created by the active site residues. So, it is perhaps more appropriate to think about this as a solvent substitution rather than desolvation. Okay. Now, let us move on to the last way in which enzyme catalysis can be understood, which is strain or distortion. So, we all know that epoxide ring opening is much faster when compared to its acyclic ether counterpart. So, the way we understand this is that the oxygen is present in a ring which is highly unstable. Once the ring is opened, it results in increased stability. Okay. So, therefore, this opening of the ring contributes to some instability in the reaction. And so, one could understand this in terms of ground state destabilization, right. So, to look at another example from a biological context, alkaline hydrolysis of phosphates, okay, phosphate diesters, is significantly faster when you restrict it into a cyclic system, okay, and the rate acceleration is about 10 power 8. Okay. So, this suggests that once you increase the energy of the ground state, it can result in a dramatic acceleration, right. So, what does an enzyme do? Okay. Enzyme creates a situation where you can have enzyme creates a situation wherein you can have strain or distortion that promotes the or increases the rate of the reaction. Okay. Now, in a nutshell, catalysis is typically achieved by ground state destabilization or transition state stabilization, and both of this will result in acceleration of rate. Now, some enzymes need the help of small molecules to catalyze reactions. So, these small molecules are known as cofactors. Okay. So, cofactors are nothing but small molecules or non-protein substances which are required for reactions to take place. Okay. Deficiency of some cofactors, typically because of malnutrition or poor diet, can result in the loss of enzyme activity, and subsequently one could fall sick. The example is scurvy, which is where vitamin C is required. Okay. Now, the knowledge of how the enzyme how the coenzyme binds to the active site, or how it catalyzes or how it enables the reaction, is very important, because if we want to develop inhibitors of these enzymes, let us say in a bacterium you want to inhibit a coenzyme, then it would be useful for us to understand how this coenzyme works. Okay. Cofactors, as I mentioned earlier, are either small metal ions or they are small organic molecules. Now, let us look at some examples of these. Okay. The first example that we could look at is NAD, which is nicotinamide adenine dinucleotide, okay, or its phosphate analog, which is called NADP. Okay. NAD is a very important cofactor, and you can see that it has a pyridinium ion as the main functional group. Now, this pyridinium ion functional group is an excellent acceptor of electrons, especially when you look at hydride. So, here you have an example of a enzyme which has a nucleophilic active site, which is reacting with an alcohol, pulls the proton from hydroxyl group, which results in migration of the electrons between the OH to form a carbonyl, and the hydride is then transferred to the pyridinium ion. So, here NAD plus becomes a neutral NADH. Okay. This enzyme that we are talking about here is lactate dehydrogenase, and the substrate for this is lactic acid. Okay. Now, NAD plus has to be bound at the active site along with lactic acid. So, in this enzyme, of lactate dehydrogenase, it is expected that you would have a binding site for lactic acid and a binding site for NAD+. Once the binding occurs, oxidation is facilitated and you form pyruvic acid, which then dissociates. Now, if we were to look at various classes of enzymes, they are found, you can find them in this table, and they are typically enzymes which carry out oxidations or reductions, which are known as oxidoreductases, or they can transfer groups. For example, glutathione, they are called transferases, and in the case of glutathione, it would be called glutathione transferase or glutathione S transferase. And then there are ones that can hydrolyze functional groups, and these are known as hydrolases. And you can also have enzymes which are lyases, which basically form double bonds, which result in loss of a group to form a double bond. And then there are ones which are basically isomerases, they convert one isomer to the other. And lastly, they are ligases, which help in forming a bond between two different substrates. And typically these are dependent on ATP, which means you need energy to do this. So, these are the major classifications of enzyme, and keep in mind that enzymes that can catalyze the forward reaction can also catalyze the reverse reaction, because it is in an equilibrium. Okay. So, an oxidase an oxidase enzyme can also do reductions. Okay. Now, once the enzyme has done its job, you would need for the enzyme to stop working after some time. Otherwise, it will just keep on consuming the substrate and giving you the product. Okay. So, enzymes have to be regulated inside the cell. One of the mechanisms by which an enzyme is regulated in the cell is called as feedback control. So, imagine that there is a biosynthetic scheme that is happening. So, you want to synthesize or there is biosynthesis of a molecule which involves n number of steps. Okay. Now, as long as a substrate A is available and enzyme 1 is available, then enzyme 1 is going to turn over A to produce B, which is then the substrate for enzyme 2, and that forms C, and so on and so forth. Now, this final substrate product G can be an inhibitor of enzyme 1. Okay. So, once the levels of A go down and levels of G go up, G starts to compete with A for the same enzyme, and it prevents the reaction from occurring. Okay. So, this is one of the ways in which enzymes are regulated. So, this is called as feedback control. The other way in which enzymes can be regulated is through allosteric regulation. Okay. Allosteric sites are nothing but sites which are not at the active site, but are in a remote location. So, here is the schematic representation. So, imagine that this is the active site of the enzyme, and here is where the substrate binds. You could have another site on the side, which is an allosteric site. And if we want to regulate the activity of this enzyme, what would need to be done is for this substrate, which is an allosteric inhibitor, to go and bind to the enzyme. Once the enzyme binds, you have a conformational change and the active site is blocked. So, previously when the substrate used to bind to the enzyme active site, since the conformation change has occurred, the substrate does not bind anymore, there is no fit, and therefore, the enzyme can be inhibited or regulated. Okay. This type of regulation can be at the start of a biosynthetic pathway, and the allosteric inhibitor can be much later in the pathway.